Welcome to this other Kepler's Garden. Kepler's Garden at Planet B in Amsterdam at the Science Park. We're broadcasting from the Science Park, and this is the stream made by the Waag team in Amsterdam. We are invited by Ars Electronica to also make a program special to this, let's say, COVID issue of Ars Electronica. Invited to represent another Kepler's Garden, which is a streamcast tonight to contemplate the idea of the garden and how we as humans relate, or should we relate, to ecology, nature. And tomorrow, another one with new guests and other sorts of reflections there. Tonight, we're going to have a conversation with artist Christaan Zwanikken and curator Alice Smits. Thereafter, we're going to experience an artistic intervention by Esmee Geerken that will lead us through microscopes. And last this evening, we're going to have a conversation with evolutionary biologist Toby Kiers and artist Saša Spačal. And it will all happen around this idea of the garden. We've tried to do this at night. As you can see, I'm somewhere where it's dark, because it's also about Kepler, the astronomer, looking from Earth into the sky and back, looking at the sky as a reflection to look at ourselves and how we relate to our surroundings. So let me first introduce quickly Christaan Zwanikken and Alice Smits. We're going to collaborate with them. There they are. Fantastic. We're going to collaborate with them in Amsterdam in a project called Smart Hybrid Forms, where Christaan is going to work together with the laser lab of Free University, but also with the organization Zone to Source, in this case represented by Alice Smits, and V2 organization for unstable media in Rotterdam, with a long track record in art science investigations, let's say. First we're going to listen to a short presentation of Christaan, who will show us a bit of his work, and thereafter tell us a little bit about what he aims to do in his Smart Hybrid Forms program, where it's about trying to get a new understanding of how we should co-inhabit the planet with plants. Not only with the ecology at large, but specifically with plants. So he will say something about that research he's going to do, and thereafter that is commented by Alice Smits of an organization, Zone to Source, that has an art program that is focusing mainly on art and ecological issues. So I want to switch to you, Christaan. I don't know whether you first want to show the video you have for us, or that you first introduce yourself further.
No, maybe it's a good idea just to have an image of what. Well, I did this exhibition in 2017. There was a kind of mixture of like the older work, and that goes into the new stuff I'm working on, which is related to plants and plant communication through chemicals, and so I think it would be nice maybe to have a little quick view at the work, and then maybe from that I can start explaining a bit, or find a shortcut to the hybrid smart hybrid forms project.
a very sensitive system that's very prone to break down.
As time passed, I began to play a game with water's secret powers. My jurisdiction is one your life, Father.
It won't just sit there and die off.
I surrendered my so-called free consciousness and allowed the water to take possession of it for a while. Little by little, this game turned into a profoundly earnest endeavor.
Senor, you know we have no money.
If you're a man we can trust.
No, we have no money.
Senor, you know we have no money.
Maybe 200 of them. Rented rooms you live. Rented rooms you live. 500.
So, hi, Christaan, you're back.
Hi.
I think that's amazing work you make. So tell us a little more about it.
Well, what do you want to know?
I also know you're setting up a huge exhibition in Germany at this very moment, so you must be tired. So let me ask you a few questions about this work.
I'm midway, so I had a week off, and it's one of my biggest. It's a 2-ton installation on the ground, so it's a lot of really physical and dirty work. And but I'm halfway. So I slightly regret to take up the invitation this late, but I think it's all for the arts, and especially in this situation we are in all right now. So I'm happy to, but if I zone out, you just keep me on track with the conversation that I don't
no problem. No problem. But let me do about questions. I see a lot of humor in work. And I also see a lot of mechanic mimicking of animals and also other organisms. So now for Smart Hybrid, you plan to make a plant or even a tree in order for us city dwellers to make us understand urban ecology better. How do you envision that is going to look like? What kind of sensors or mechanics?
I have no idea. I have a lot of ideas, but in this specific case, I mean, we haven't started yet. I haven't started yet at the university. I haven't met the people yet. Of course, we had all the Zooms, but that's all, that doesn't work. So I haven't been at the university yet. Yeah, I just want maybe 10 minutes before COVID-19. So, and of course, the whole situation actually is, of course, influencing a lot of the ideas I basically first had.
But tell us a bit about the ideas.
Well, there's many ideas, but yeah, it's so hard for me to. You say it's the mimicking of animals, and so that has always been the problem a bit. That if you portray, if you work with animal remains, or that it is seen as mimicking, and that's actually how I eventually got to plants. But it's a really long story, because actually it's a lifelong time of experimentation and observation, which first started with animals when I was a child, actually. And I had like 3 things that were like fascinated me completely. That was animals, there was technology. My grandfather was an airplane technician, and I felt like creating these mechanical devices or technical devices or craftsmanship put me in this transcendental state experience, which I found very interesting already as a kid. And then there was this music and sound interest, and I think this came. I was like born really like, my mother was a ballet dancer. I was born with music and sound. So those things always fascinated me already as a child. And so first I had all these animals. I had all kinds of pets, and of course dogs, cats, and I also would like, I did experiments with them, but not like horrible things, just to see if they were sentient, if they could smell, what they could see, what they prefer to eat, and why they would and so I had also like birds, and eventually I had a chameleon, so a reptile, that I had for about 5 or 6 years. And I had this really strong connection with this reptile, because I found out that actually he could see me all the time, because a chameleon can see three dimensions all the time. But I was curious if he could like smell me or hear me. So I did first test like trying to get his attention, my voice. And a chameleon, and I also had to like, I would bring him outside to have his dinner because I had like these cages where I would feed him crickets. I was completely fascinated by crickets because I had to buy crickets at the pet shop. So all the time I got fascinated by this, but the chameleon, I found out that actually he could hear me. So I could leave him outside. He could just have his own dinner, like catch flies in summer especially. And I would be able to call him, and he would come back, crawl up my arm and on my shoulder. So these were like very profound first, how do you call this? Well, experience with sentient life forms or intelligent life forms. So this has been going on all my life. I still experiment a lot. So from this experimentation, when I was very young, I went to art school and I studied sculpture. But I thought it would be really nice to have a kind of, interactive or behavioral machine. So not kinetic art, not in the sense of kinetic art like it was like this more. It's about the form of the movement. For me, I was most interested in behavioral artworks. But I started in the late 80s, so there was no digital means to do that. So it was all analog. So what I did, there was this shops in Holland called Tandy. They were, from America, but they sold these guides, and catalogs with integrated circuits. And I just bought these integrated circuits and then, tried to find any. I was horrible at it, like, I didn't understand anything of like electronics or whatever. But eventually, slowly, I started to connect all these integrated circuits, and I was able to make kind of autonomous systems. And in combination, like with mechanical systems, like really trying to create some kind of new life. And this was all very intuitive. And of course, later in the 90s, there came the microcontrollers that made things much, they're not easier, but it made it possible to do even more incredible things in sculpture. But it was always actually an idea, maybe the idea was that actually life is for like a great part mechanical. We are mechanical devices. And someone who found that out early on was Leonardo da Vinci. He stated that life is based on mechanical systems. So would there be any difference between a machine and an organic organism that moves? Even the most simple organisms have like small mechanisms that make them move around. So what's
Christian,
really different?
Here I have an interesting question for you, because from this mechanical understanding, how do you move to plants and to build plants?
Yeah, that's why I try to make it shortcut, because it's a long story. Because then, of course, like in the early 90s, robotics in art were really new. You had like Mark Pauline and Jim Whiting doing all this crazy stuff, using military equipment, and they got hands, or they stole it or something, and they did these incredible things in the early 80s. So I was very influenced by them. So how do you get to plants? Well, it's the same observation. It actually came. I've always been working with plants. My grandfather, my parents were like devoted gardeners. I was actually always a very, very devoted gardener. Not that young, but early on I started to, what's enter, because in English enter to glue two pieces of like trees together, and you can, I learned all this stuff in Portugal. My parents moved to Portugal and we came as me and my brother. They're very young, like in a nowhere place, and there was only nature and so on. So people that told us you can only how to grow things, and to take cuttings from one tree and you can glue like an almond tree and you can put a fruit tree on top of that. And then, so there's this technologies of using plants to disseminate or to grow trees. So all these technologies I already learned as very young, and I was extremely fascinated by that. So that you could grow this stuff or create these kind of trees or get kind of specific tastes from trees. But in that process, and that was completely out of like any art thing, I saw this amazing things in nature. I saw plants doing things that were amazing, that they seemed to know exactly the direction where they needed to grow to grab onto something. You know, if you look at vines and so on, and but many other experiences. But I always found this because I grew up there. It was just normal. It was, and it's so. So I had all this experience already, and I did a work like maybe 6 years ago. I really tried to make synergetic work, so that it really um, triggers all the senses. And I think it's interesting to see how far you can get with like triggering all the senses before you get to a kind of even more transcendental. First, use all the senses, and so I never touched smell. So I thought I think I always did a little bit of smell installations. I thought I want to do this like different. Um, so I created this work, it's a bit in the video, that's called Nose Patrol. That's actually a computer-controlled perfume generator. So in real time, it produces perfumes. But for the perfumes, I thought it would be really important to use the real smell of the plant. So essential oils from the real plant, because perfumes are in fact synthetic. And I did a lot of research that synthetic smell is quite harmful for our, limbic and system, and for many other things. Because so I thought, and we are bombarded with like synthetic smells. I thought it would be nice to make two cannons that would bombard you with kind of healthy smells.
So
And for that,
are you going to work with smells again?
I don't know. Yeah, I mean, yes, but so that got me into the whole chemical world of plants, because that's how they communicate. For like, probably that's their language. What we think that it smells nice, but there's a purpose, because plants don't spill any energy in producing those essential oils. So it is something they need for something. And we all know, science knows it's like it's a kind of chemical warfare to protecting from diseases, from pests, from insects, also from the heat. So there's a lot of there's still a lot of
there's still a lot of things we don't know there.
Well,
I'm going to ask you a question.
Most we don't know.
Yeah,
we don't know anything.
And is it also because I want to switch to what Alice can add to this conversation? Is it also really a need for you to focus more on plants from an artistic point of view, but also from the view that we as humanity need to relate to plants?
Oh yeah, it's for me, not a question, or for Alice.
No, it's for you. That's a question.
Okay. Well, I think that there is really the experimentation and the observation point that is probably half of science has been neglected enormously. Yeah. And I can only say this because I looked into things, I tried to get information, didn't get it scientific, and I just started to experiment myself. I will not get into the details because I think also we'll save them a little bit for the projects I will present later. But this has brought me to very interesting things that by experimentation and observation and simply writing it down and do it again and again, and I find that completely fascinating. And the whole chemical part, that's something I really would like to develop at the universe, or to research on at the university. And
Great.
yeah.
That raises quite some expectations there. I'm very curious where that's going to lead you. I now want to give the word to Alice, because I think there's a very interesting bridge here, because you say a lot of the synthetic olfactory elements in the air are environment. Point of view that you find also in a lot of the artworks that Alice is about zone to source, and what your interest from the zone to source perspective is in joining this research that, among others, Christaan is going to set out in the Smart Hybrid Forms project.
Yeah, okay. Yes, just like with a brief presentation about what I'm doing in the Amstel Park at Zone to Source. It's actually a project that was very much inspired by the park itself, which you see now on the picture at the Amstel Park in Amsterdam. Was originally a place for a horticultural exhibition, a World Expo on horticulture. And in 2013, I started this project and really got inspired to really rethink what a horticulture expo could be, and what this new perspectives could be about how we relate to nature, and how we can rethink relations between human nature and technology, and through artistic imagination. So since then, over 6 years, I've started a project here that is very much thematically driven. And I'm inviting artists who, from many different kinds of perspectives, are exploring new relations, new experiences, new practices between humans and nature, and how we can start reconnecting and reflecting again on what our position is as humans in the multi-species world that we are living in. And so it's as a curatorial project, it's also very much a research project. And for me, it's always very much important to keep research and curatorial practice together. And we very much use the park as a kind of living lab. It's a municipality park where we're trying to do as much as we can, also doing workshops, have artists work there, trying slowly also to see if we can continue this more as a park for art and ecology. So this is the space that we're working from, just to show you a few, give you a few images of what it looks like. This is an orangery, a glass house that we're using in the summertime. It was a project by Sonja Baumel, 50% Human, which was a really interesting project that was reflecting on the fact that we as humans only consist of 50% human DNA. So we already are kind of an ecosystem ourselves, which completely changes our notions of what actually the human subject is. And thinking about autonomy and unity completely changes when we think about ourselves as a complex habitat for other creatures as well. And this was an installation that was trying to make us reconnect to these aliens that are part of our own bodies. We also do a lot of projects outdoors in the park. And this, for example, is a bus bench by Annemie van Splunter. It's a project that's been in the park for 5 years, which is a space where humans and bees can be together. So it's at the same time a hotel for bees, as well as a place where humans can hang out. This is a project which is recently brought into the park by Ronald van der Meijs, Oroshi Cloud Sequencer. It's a sound project. It's completely functioning through natural phenomena like the water, the wind, and the sun. So when there's no sun, there's no sounds, but as soon as the sun comes out, the pumps are filling the water and sounds are gonna appear from these instruments. So it's a really project that's also about how we can get different attention, and how we can not expect immediate consumption, but really experience ourselves through different kind of time zones from the time of natural elements. This was a project by Jos Volkers, a bioremedying missile that just left the Amstel Park, and it's placed very much with these. It's a seed bomb. It's the world's largest seed bomb, completely filled with seeds from Romania. And it's a project that reflects a lot on technology and nature, and it puts all these kind of blunt questions, whether we should actually go to space? Should we stay on Earth? Should we blow ourselves to pieces? Or in times of ecological catastrophe, maybe explode a big seed bomb on Earth and do something about our home habitats. And then we use the park as much as we can, also for research. And these are some pictures of artists at work, but we do a lot of events. Like the right picture, we went with a geologist and we made a bore drill 10 meters down the earth because we wanted to know what the earth and the soil was like in the park. And then we have discussions around this where we're looking at this kind of historical narratives. This was a project by Jacqueline Mierema. Left is the Onkruidenier. I'm working a lot with them. Also in the next years, we're working a lot on the park to start telling these cultural stories around plants and how you can think of migrations of both plants and humans together. In the next couple of years, we're making up a plan to continue that and make it also a more readable park where people actually can encounter it in this Floriade park, how people and plants are, what the different cultural practices are around plants. This was a project by Cecil Mariton and Jonathan Rose, Cartographies of Human Sensations. This was also a kind of performance pieces that we are doing in the park where we work together with people, which are very much about how we can attune ourselves different, what kind of strategies of attunement can we have to nature. And this worked a lot with the data, both data from our bodies as data from the environment. And how can we kind of connect it and see our bodies as sensors in a way to sense our environment in different ways. This is a project Machine Wilderness that I've started in 2015 for several years together with Teun Karelsen. And it's a project that is kind of rethinking the relationship of designing technology in relationship to ecosystems. How can we think not of human-centered technology, but how can technology really first live within ecosystems and be supportive to ecosystems as a whole. And this is, I think, a good example also about the way I try to work in the Amstel Park. We very much try to make a space for experiment where artists can work, experiment with new ideas and also be present in the park and at the same time also interact with audiences because we're in a city park. So we get people who come especially for our projects, but then also a lot of people who might never come to this kind of exhibitions, just drop by because they leisurely walking in the park. And so this was also a project where we had 5 artists working for 2 months in the park, working on new robotic projects that were relating to the habitants of the park, animals and plants in the park. Also testing experiment things outdoors. This is Ian Ingram, who's working one of his robotic experiments. Another project like this, Ivan Henrique's Symbiotic Machine, we did that very early on. It's a project actually that was developed at Zoongestos and then that year won a prize at Ars Electronica. We did this together with Raoul Vreese. Ivan Henrique worked with Raoul Vreese, who is also the person behind the Hybrid Forms program. And Ivan and Raoul worked for a year together as an artist and a scientist. Working on the Symbiotic Machine, and this was a project where we built a very big pond inside of the glass house. And for 2 months, we had Ivan with Raoul and with students and other scientists working with Raoul, experimenting on this project. And it was a robot that could detect algae that were set out in the water, swim to the sunlight, and through photosynthesis was again gaining energy to continue swimming. So for 2 months, we had a lot of workshops around it, both with children as with students and other people interested. And for 2 months, it worked. And the very last day, it actually exploded. So that was a very interesting ending.
Yeah, it's very interesting to know that Ivan Henriques has presented this very evening at FACT in Liverpool as part of another Kepler's Garden program that is also streamed live today.
Okay.
Yeah. Also looking a little bit at time, it's great to have you here to see all these projects. It's interesting to see that Christiaan's ideas around working with plants and the sense, the olfactory elements, the volatile elements they spread is very interesting because we tend only to understand them as a nice flowery scent, but not all the other parts of the spectrum that are also there. I don't know whether you have had any earlier works that look at that same aspect of ecology, Alice. Did you?
Yes, we actually have done quite some projects that really deal with this different kind of sensory experiences. Also, a few years ago, we did Project Secret Signals, which was very much about that, not just smell, but also about hearing, about feeling, also how we can make sounds, for example, more tactile through vibrations, and how we actually use our bodies in a way, as in that sense, as sensors to experience differently. Because of course, smell, for example, is very important in our social relationship and now moving through the world. But we have very little, we barely have a language for it, or very little conscious about it. So a lot of the projects we're doing actually deal on that level. And it's great to also use a park because we're also working outdoors. And I think in that sense, I use the word attunement, but I think it's very much about different kind of tuning strategies, how we tune in differently. And we also, I'm developing together with a colleague of mine a project for the next 4 years where we want to build a school for interspecies knowledges, from a desire to work much more profoundly for longer terms together with people in the park and really, really think what all this kind of new knowledge, all this new experience really mean for the way we build communities, what an interspecies community could be, and what does it mean for our way of working and our way of living together, and really thinking through these notions and what kind of tactics. That's I guess what I'm trying to say, to really think about the tactical level. How we can exercise that together. So it's a kind of training in that sense as well. I always like that. Think about it. Like we almost use art now. There's a lot of training ideas going on in this kind
I have one last question for this session to the two of you. As this is a program that is titled Kepler's Garden, so it's about contemplating the idea of the garden, where humans control or tame nature. Can both of you give me an example of the opposite, where you think that nature is taming us, or other species are gardening us?
Well, I think there's always a very good example that I always like about grain. I mean, we can say when people started domesticating and started living, not being nomadic and getting themselves into farms and starting managing the grounds and soil around them. But I think more and more you also hear the argument that really grain has been domesticated humans rather than the other way around. One of the most successful species nowadays is grain, whereas humans are slaving away. And we think that finally sets us free, but we're only slaving away more on our farms and in our factories to get this grain to grow all over the world. So I think this is already a kind of different shift of thinking, and who is domesticating human, who is in charge, so he's master and servant.
Yeah, but this is actually, then we are talking about nature before this incredible collapse of biodiversity we have at the moment. So I think there's no, I think the real problem is that it's like easy. It's not this is not my quote, but it's James Lovelock, who the Gaia theory scientist, that says, he has been planting trees his whole life long because he thought, at least I can plant trees. But he says, you cannot plant an ecosystem. And that's really where the problem, we cannot create ecosystems. And we know very little about those ecosystems and the complexity of it. And it's actually so complex in an ecosystem that's even probably hard for current science to put this in any kind of model. And so this is really, with this collapse we're seeing now, what are we going to do? I think there are some ways to help ecologies but we have not the slightest idea how they exactly work. It's just too complex. And I think scientists, artists, farmers, scholar people, non-scholar people should actually through programs like I'm in now and many others, to see if we can figure out how to sustain those ecosystems. Because it's just a crazy idea that maybe, the United Nations calculate that like in 20 years, 60% of the world population will be living in cities, completely disconnected from biodiversity and from any ecosystem. So how are we going to manage that? And so we need a kind of complete new methodologies to do something. And I think we can still shift things. I don't think there's like dystopian world right ahead, but I think we really need to get into this. Yeah.
So it's interesting if you turn the logic around of weeds or domesticated farm animals actually being evolutionary very successful because of us. They must
but they don't need technology.
No, but
so
they
we are using technology. Nature doesn't need technology. It just does everything on its own, and you don't need to have any worries. And there's many examples of
but
how
you can domesticate animals without technology. So, but if they domesticate us in order to be evolutionary successful, you can also say that the garden is gardening us, and that I think I want to round off here in order to give the floor to Esmee, because I think it's interesting what you say about us being detached from understanding what ecology is, as we are living with higher and higher numbers in urban areas. So there's a even bigger need for your kind of work to be given a stage in our cities in order to not lose that connection.
Yes, but there's actually a wrong line of thinking in this. What I thought before, that you can use like these areas in cities like zoos, parks, maybe gardens, to have this connection. Still have, especially like for young people and children, that there's still this connection to biodiversity. I think this is essential for people, but you cannot create an ecosystem.
Yeah,
you cannot get all the insects and the spiders and everything that does the trick.
Yeah.
So there's still
a lot more to be discussed, and I'm very much looking forward to starting this collaboration with you, with University and Raoul Vreese, with V2, and with Zone to Source.
I think it will be interesting. Yeah.
Super. Hey, I want to thank
about these new ecosystems we're going to create with these new entities and hybrids. Is very much part of our program.
Yeah. Thank you much for now. I'm going to ask the people behind the scenes to switch to Esmee, who's also going to lead us to another colleague. Yeah. So we are doing this program from Waag's Planet B location, where Esmee Geerken is currently an artist in residence. And she's also working with Marloes Bistervels, who you see in the bottom screen, who is a scientist working at AMOLF. I don't know where the abbreviation AMOLF stands for, but they do fundamental research on materials. So what Marloes is very good at is creating microscopic but very unnatural crystals. Am I right? So very synthetic crystals. And we're going to connect to her electron microscope in a bit. But first, I'm going to give the word to Esmee, who's going to lead this this whole session.
So our first common ancestor that came out of the sea 380 million years ago is thought to be the Tiktaalik. And maybe it's because we are descendants of fish that when we imagine ourselves an aquatic life, a life in the sea, we depict ourselves as mermaids. I like to question, however, if we can also imagine ourselves as human crustaceans or mollusk maids, perhaps. Creatures with exoskeletons and shells. And I wonder how they would look like. So during the lockdown, 3 businesses did very well. It were the supermarkets, the garden centers, and the DIY shops like Praxis. And perhaps this is because these shops reflect 3 human basic needs: food, some form of greenery, being surrounded by a bit of green, and the need for a shelter. Nowadays, most people don't grow their own food. At least I don't, and I also don't build my own house. But I was wondering if this maybe is making us slightly unhappy. What if the need for a shelter also comes with an urge to build one yourself? So if constructing a shelter is in some way still part of our DNA, I wondered how, if we reconnect to this DNA, how would these buildings look like? So could we, for instance, build like corals or build like the common ancestor of actually all animals, the mineralizing sea sponge? Maybe these creatures can re-inspire our building strategies. So humans appear not to be very comfortable with their nakedness, a feature that they share with shellfish and snails. If you think about the shelters humans create, you might think about 2 categories. The first one is garments or clothes. So the word garment comes from the French word garnier, which is derived from the Germanic garnish, and it means to cover. So clothes cover your nakedness and provide some kind of portable thermal regulation. And they're often made from organic materials. And if I talk about organic materials during this talk, I mean materials made from carbon molecules, so made by organisms. Materials like silk, wool, cotton, fibers, and plastics. The second category of shelters is, of course, the house or the building. The word house comes from the Old English word husan, which is probably related to the root of the word hide. And houses, they're mostly portable walled protections against weather influences. They indeed protect you from the gaze of other people. These days, buildings and houses are usually made from mineral components, such as cement, which in fact is made from limestone, and bricks made from clay, another mineral. So we knit, we weave, we sew, we scaffold, we cement, we lay bricks, we pour concrete, all just to cover ourselves and to hide. I became interested in what we can actually learn from the buildings of other creatures, such as shells and mollusks and vertebrates that create skeletons and shells and eggs and bones. And specifically, I was interested in the building techniques of foraminifera, because I've just spent 4 years doing my PhD about them. So I learned a lot about their building strategies. Foraminifera are unicellular marine organisms that secrete a calcite shell by precipitating it on top of an organic template. And these materials, they are very remarkable exactly because what they do is they combine the organic and the mineral. And this results in giving them these very intricate shapes that are only possible if you have material that is very strong, but also flexible, and that has optical properties and all these amazing features that I thought, oh, what if we could actually use such materials as our building materials? And I thought, what if we relearn how to calcify again and how to agglutinate our houses and become crustaceans or shell-bearing species again? So here in the Planet B lab, as you can see here, I'm experimenting with creating such hybrid materials inspired by the shells of foraminifera that you see here. And I'm trying to create sort of these hybrid materials. And out of these hybrid materials, they inspire me to see how an exoskeleton could look like. So I'm combining the organic, the clothing, with the mineral or housing. And in one of the experiments I recently did, I was making a hydrogel from gelatin. So that's an organic component actually coming from your bones, but it's still the organic tissue that sort of holds your bones together. And I had some discussions with a scientist, Lucas Helmbrecht from AMOF, and he suggested that what if I put the salts that I have, so I'm working with calcium chloride and sodium bicarbonate, what if I put them inside the jelly? So that's what I did here, and I let it react. And then I prepared some stubs. So I did many different experiments trying to mimic shell formation in different ways. And I cut out little pieces and put them on these scanning electron microscope stubs, took them to AMOF, just 500 meters away from here. And then we were going to look at what actually happened. And to our surprise, we saw these spheres, these microspheres. So the spheres are just 2 micrometers in diameter. And what was actually even more cool was that they're actually hollow from the inside as well. And if you think about it, that's very interesting, because normally if you would grow calcite just from a solution like seawater without any organic or any life interacting with it, you would get these cubical or rhombohedral, as they call it, shapes. So that was very magical to see. Let me just check, is Amel still off? Okay. These experiments actually made me think about what if we place our building materials in a biogeochemical perspective? How does that change our perception of building materials? So lately we talk a lot about circularity, but what if you look at circularity from a geological timescale? So if you look at limestone, which is the main ingredient of cement, it actually consists of foraminifera and coccolithophores. So coccolithophores are another type of calcifying organisms. And these organisms that make these microscopic shells, they originated 500 million years ago. And since then, they have been sinking down on the seafloor, being compacted and uplifted into rocks. And then we take these rocks and crush them and form cement out of it. So I thought, yeah, if you know that ore houses are actually made from the houses or shells of these microscopic creatures, what can we learn from how they make their shell? And can we maybe in some way shortcut this geological timescale that is involved in making limestone? So, you know, perhaps it could be a bit faster than taking 500 million years. So that's what I tried. And I'm trying to, how I'm trying to mimic it is by seeking what kind of organic materials I can combine with calcite, or how to grow calcite. And I'm doing this in the project called Build Like a Shell. In this project, I'm trying out various ways to mimic what they call the privileged space. So the privileged space is a secluded space that biomineralizing organisms form, organisms such as mollusks or foraminifera or other shells. They create a space where they form these kind of organic tissues or a template, and then they mineralize it. And that's what I wanted to try. And for this, I'm collaborating with many material scientists who actually already know a lot about this, and also designers and artists. So during my PhD, when I was looking at foraminifera, we were trying to find out how fast foraminifera actually grow their shell. So I went to Japan and did an experiment there. And then we figured out that they grow their shell at a speed of 0.5 micrometer per hour. So then if you do a little calculation, actually it takes a year to just simply precipitate a kitchen tile of 0.5 centimeter. So basically that is what I was trying here in this image. I had a bit of hemp paper, so that's these lines that you're seeing. And then I put it in an aquarium mimicking this privileged space. And then I tried to precipitate minerals on top of it. But actually, it was very frustrating, of course, with how long it takes to precipitate calcite yourself, and how difficult it actually is to mimic something such very amazingly difficult and beautiful, such a shell formation in a simple lab, such as the Planet B container, where I don't even have running water at the moment. But then, in a way, dealing with this process of trying to grow shell material myself and seeing how slowly this actually is going did make me appreciate a lot more these bricks of cement, that if you realize that a brick like this, how many shells it contains and how much years it took to build these shells and to precipitate them all on the sea floor, that I started to appreciate just a single block of limestone, a brick of limestone, a lot more. So a lot of limestone was formed in the Cretaceous, a geological period 145 to 66 million years ago. And during this time, you have to imagine that it was very warm. So sea levels were much higher than today. And if sea levels are higher, then you can imagine that a lot of the land is actually flooded with these shallow, warm, carbonated seas. And these kind of seas provide the perfect habitat for biomineralizing organisms to form many, many shells. So this is a period when, for instance, the White Cliffs of Dover were formed. And if I think cement was originally, well, in some modern form, the Portland cement that is actually quite polluting was made in England using these limestones. And if you think about how that we just crush all these shells and then mix it with fly ash, a byproduct from coal combustion, and then create a material that, if you think about it, is just very hard and brittle. It needs iron bars to kind of support some kind of strength. And then we sort of pour it over the entire planet. So cement and plastics, these are the materials we most widely use these days to build our products and our houses. So you could say that they're actually not very elegant materials, as they pollute or they actually disrupt biogeochemical cycles. For instance, when producing cement, a lot of CO2 is produced, so disrupts the climate system. And plastics, of course, falls apart into these microplastics that pollute the oceans. And these are materials we kind of rationally use to design our surroundings. But then if you think about our bodies actually can make very elegant materials. Our bodies do know how to biomineralize. So our bones are really wonderful materials made out of collagen and a mineral called hydroxyapatite. So how can we move towards a more biologically intuitive way of building with minerals? For this, I'm collaborating with the self-organizing matter group at AMOF, and they found a way to create this really amazing shapes out of liquids. So Wim Noordijn is the group leader of this group, and they make these kinds of shapes. And what's very interesting is that they can control the shape, but also the composition of the material. And if you compare this to how we make cement, it's just really great that you can have this type of control on just the nanoscale, forming these very special shapes. Other scientists I'm working with are researching bone material, and I'm also very interested to work with scientists that are able to create mother of pearl. Partly, so there are people that developed a way to create mother of pearl using mineralizing bacteria and urea. So astronauts could actually do this themselves on the moon using their own pee. But for today, what you've just been looking at partly, I think I heard at some point there were maybe some problems. You've been looking at the nucleations of crystals in the lab just 500 meters away from here. So I'm happy to introduce to you Marloes. And she is a scientist at AMOF that is creating these shapes and has been growing live now on her free Friday night these shapes for you, which is actually, I don't know if you realize it, but it's very special to see these crystals live under a microscope. So I hope you enjoyed it. For the last, I would really like to thank Marloes and also all my collaborators in this project. So the Onkruidenier, the self-organizing matter group of AMOF, my scientific advisors, Aqua Minerals, and also, of course, Laag for hosting this great evening, Lucas, Micha, and Chris. Okay, thank you very much.
We're back after this intervention by artist and chemist Esmee Geerken. And we're now going to talk with an amazing scientist, microbiologist, Professor Toby Kiers, who will present her work, and audio and bioartist, correct me if I'm wrong, Saša Spačal from Ljubljana, who, despite Corona limitations, has been working a lot in Amsterdam the last year, because she was supported by the 3-package deal possibility of the Amsterdam Fund for the Arts, which Toby, me at Waag, and Willem Veldhoven of Mediamatic share every year. So every year we are allowed to select a new artist, which is fantastic. And I think this selection of Saša to work with us was also fantastic. It's interesting to see the relation between the works of the research of Toby Kiers and the work of Saša Spačal, also in the frame of this Kepler's garden here, where we talk about many different possible meanings of physical and metaphorical gardens, that of our planet, that of the science park where this all happens, the garden that we have with our houses, but also gardening as such, both as a research activity, maybe a very political activity, also an activity where we have a lot of relations with other species. And that is also a little bit where we're going to have a discussion about the way other species interact, that show similarities but also differences with how we humans act among ourselves, barter, deal, exchange, and so forth. So I first want to give the word to Toby, who is going to show us a few fantastic pictures about her work and tell very briefly about her amazing research in microorganisms, in fungi, etc. So please, Toby, the floor is yours. Please share your screen.
Great, thanks so much, Lucas. Real honor to be here, to be able to speak about the things that we are studying. Most of what we're doing right now is work underground, and I think a lot of us don't really have a grasp of what the underground looks like or what's going on down there, and so I'm really excited to sort of share some of that with you. So I'm going to share my screen. Okay, looks like it's full screen now. Great, so what we do in my group is we study the evolution of cooperation. We're really interested in how organisms form partnerships in nature, and how these partnerships can sort of withstand the test of time. But actually what we're even more interested in is conflict, and basically trying to understand how conflict drives the breakdown of these partnerships. We work on the basis of natural selection. I'm trained as an evolutionary biologist. And of course, there's this idea that if you can gain a little bit more from a partnership, then you're going to tend to spread. You're going to have your genotype spread even further. And so on the one hand, we see all these beautiful cooperative partnerships in nature. You can even think of things like plants and pollinators, all kinds of different what we call symbioses. Yet underlying all those beautiful partnerships is this conflict of this idea that what if a partner took a little bit more than its quote unquote fair share? And so one of the partnerships that we're most interested in studying these kind of cooperative and conflictual ideas is in one of the most ubiquitous symbioses on Earth. And that's the symbiosis between arbuscular mycorrhizal fungi, it's a huge, big word, and plant roots. So basically, wherever you walk, you're walking on these beautiful, huge networks of fungi and roots. And the fungi, this is the picture that you see in front of your screen, are the very white, fine filaments, and the roots are the more yellow-colored ones. And in this symbiosis, the fungi actually colonize inside the root cells of these plants. And at that site, they exchange nutrients. And so the plant is feeding the fungi carbon, sugars and lipids and all the things that the fungi need to grow. And in exchange, the fungi is collecting resources out in the soil, like phosphorus and nitrogen, and trading those to the host. And what you see in this symbiosis is you've got a fungi that can connect multiple plants simultaneously. And you've got multiple plants above ground that can be colonized by all different types of these fungi. So I'm going to show a very short video that kind of shows how we think this symbiosis is working. And we call it the underground marketplace. So in this marketplace, you have the roots, and the roots are what give the sugars to the fungi. And you also have the fungi, which collect the nutrients, the phosphorus and the nitrogen for trading. So these are all the fungi that are in the soil making connections with the roots. This is happening as we're walking on the ground. And here you have a fungi that connects with a particular host. Now what we want to understand is how does it trade its resources? How does it decide where to give phosphorus? Who does it decide to partner with? And what we find is that the fungus and the plants are both really good at detecting good and bad partners. So here you have a trade with very little fungus, very little phosphorus on one side, and a successful trade on the other because the fungus is offering more in exchange. Now we see the opposite with the fungus. Let's say it's connected to two roots simultaneously. Where is it going to give its phosphorus? What we find when we do all kinds of experiments is that the fungus can somehow tell a good host from a bad host and gives more phosphorus to the plant that gives it more carbon in exchange. So what this creates is sort of a marketplace whereby the fungi are connecting multiple plants simultaneously, but only trading nutrients with the ones that are giving them the most carbon in exchange. And in return, we see that the plants are really supporting the fungi that are only giving them the most nutrients. So when we first started working with this system, it came as quite a shock for many people. One, because I think people tend to think of nature as one big harmonious system. And we sort of shocked the world by saying, well, it might not be quite as harmonious as you think, because what is trying to happen by both organisms is that they're basically trying to get the best trade deal they can. And many articles came out about the soils showing a market dynamic, the ruthless side of soils, as they called it. And that if we look very closely at the relationship between plants and fungi, that we see sort of a cutthroat marketplace rather than this big harmonious system. And I think the word ruthless is a bit overspun. Instead, I would use the word practical, right? These organisms, both the plants and the fungi, have been selected over millions of years of evolution to really evolve these strategies to optimize these trade processes. And so rather than it being ruthless, it's basically a practical solution. Why would you share resources with a plant, for instance, that is not giving very much in return? So through this work, and this has been going on now in our lab for actually about a decade, we've shown some really interesting things about how these underground trade networks work. And that we see that both the plants and the fungi, if we give them all sorts of tests about who they choose to trade with, that they can really discriminate between good and bad partners. And that the supply and demand of the resources actually influence how those resources are traded. The problem is that in many things, the most interesting parts of science are hidden. So it's been very difficult for us to actually view how these resources are traded. And I actually liken it to a poker game where there's very good players of poker at the table, and they all get dealt a certain hand. And the experiments that we've been doing now, we deal both the fungi and the plants a really interesting hand. But then what we do is we basically just shut off the light and then turn it on again, and one of the partners has won. But we never get to see how they do that. How do they trade? Because we've been unable to visualize the actual trade that's going on underground. So even though we've got the best poker players in the world, we really can't see how they're making it work. So one of the goals that we've had in our lab for about the last 3 or 4 years is to try to visualize trade. This is such an important process. These are the nutrients that sort of drive all of the ecosystem processes that you see above ground happening below ground. But how can we see them? And so we developed this very cool technology where we could actually attach these quantum dots, as they're called, to nutrients. And quantum dots are these semi-conducting nanoparticles. And basically what they do is they fluoresce in very bright and pure, precise colors when they're hit with a UV light. So our approach was, what if we tag the nutrients with this nanoparticle? Can we watch how the different partners actually trade resources? So these are the kinds of experiments that we're doing right now. Here you can see on the C panel, you can see the actual quantum dot tagged phosphorus and it getting into the hyphae. And the kinds of questions that we're asking with these is to sort of see, well, how do non-cognitive organisms like fungi and plants, how do they deal with really unique economic systems? So for instance, one of the things we're really interested in is inequality, right? This is one of the major problems of our time. And we're quite interested to see, okay, well, if you have an organism that doesn't have a central nervous system, how does it deal with something like resource inequality? So we did an experiment where we compared different treatments where we either had resources that were very evenly spread across a fungal network, or we made one side very rich and the other side of the network very poor. And we wanted to see, how does the fungi respond to these kinds of conditions? And what we found was quite interesting. We found that when we increased this level of inequality, that it actually simulated resource sharing across the network, that the rich side of the network was sharing resources over into the poor side of the network. Now, of course, you could think, oh, this is just socialism at its best, right? The rich are sharing with the poor, and that's how nature works. Well, unfortunately, I think it's a little bit different than that. What we found was that it's not necessarily that the fungus was trying to help the poor side of the network, but rather, if the fungus was able to move the resources to an area where there were very few resources, the plant was actually willing to pay more for those resources. So there was a higher demand, and the plant was willing to pay more. So again, it was this incredibly sophisticated strategy that we saw evolving in a fungus, whereby it would first move resources from an area of rich abundance to poor so that it could get a higher price for those nutrients. So again, why would the fungus be moving these resources? Basically to gain a better price for those resources. So what we see is that organisms, and this is kind of a new emerging field that we call social microbiology, and it's basically trying to understand how organisms that don't have a brain organize the way they move and share resources. Now, of course, when this first work came out, it again made quite a stir. There was an article in the Economist because it said, okay, so fungi, as it turns out, are canny traders of nutrients to plants, right? When the phosphates are scarce, their price goes up. And so for us, again, this is quite interesting to try to understand, well, what's really happening in these underground marketplaces. But it also opens up even larger questions about this idea of living algorithms. And that's this idea that fungi are somehow able to sense different conditions across their network, integrate that information, and then make trade decisions. So how that is done is really, really unknown. And that's the kind of thing that we're working on now. This is just a very small video of the kinds of things that we're seeing underground. These are the nutrients that are actually moving in these networks that we are walking on. These are where the trade deals are happening. And here's an even closer up vision. And one thing I want you to notice about this is quite interesting, is that you see these beautiful flows, but then they change directions, which is really dramatic, because it means that somehow the conditions are changing, which changes the physical, the biophysical, the chemical conditions within these pipes that make the nutrients flow a different direction. So what we're focused on now is trying to predict those types of flows and changes. To be able to do that, we need to actually understand the physical architecture of the network. So here's a video of what these kinds of networks look like in a 3D slice. So imagine you're growing a fungal network that's connected to multiple plants, and that you take very, very thin slices of that network. We need to kind of be able to quantify what that looks like, because these are the physical trade routes, right? So it's one thing we can guess and say, maybe it's moving it over there to get a better price, but until we understand the sort of the physicality of that trade route, we can't make the predictions we need to make. So we're building in our group right now an imaging robot that basically allows us to segment every single node of growth in these networks and then project it in a 3D way so that we can understand those physical trade routes and see how those resources are made, and then make predictions about what a fungus should do under a specific condition, and see, is that following our predictions from, let's say, classical economics. So that's my presentation for now. Thank you.
Amazing. Amazing also to see the density of the network that you take from a very small part of a petri dish.
Exactly.
And then understanding that in every pipe, flows are going both ways. And what I was able to see a time I was visiting the lab with you and Saša, you even had a better close-up where you could see that in one pipe, flows were going up and down at the same time. That was, yeah.
Yeah, this is one thing that's really important that makes these fungi, these symbiotic fungi, even different from other kinds of fungi, is that because they are constantly trading with a host, they need to move carbon one direction and nutrients the other. And so even within this pipe system, they've evolved a way to make that happen. And we're trying to understand sort of the biophysics underlying how there can have this simultaneously bidirectional flow.
Yeah.
Yeah.
Wow. Wow. It's interesting to understand that we also have artists that have an interest in those same mechanisms and how we can understand them, what metaphors may be used to understand them more profoundly, more deeply. So in a way, well, for that I would give the word to Saša, who has been working in Amsterdam, but you all also much earlier have been working with microorganisms, but also with insects, etc., understanding their agency within an ecology. So I'm also very curious about to hear more about your work, and then to see whether those metaphors of economic exchange, of marketplaces, how these can be discussed and what we can learn from Toby and your work. So please, Saša, show us some more about what you've been doing the last year in Amsterdam, but also maybe earlier. I'm curious.
Yeah, I'm going to talk about a bit earlier also. And thank you for invitation. And thank you for just having this opportunity to talk to Toby again, because it was a very exciting year and a lot learned. So I will just start sharing screen and show. So, sorry. Okay.
Yep. That works. Yeah.
No. It's going the wrong direction, I'm sorry. Okay, so as an introduction, I would like to say that in my work, I'm interested in a very particular subject. it's not an iconic organism or a specific landscape, but it's more like planetary metabolisms and its entangled feedback loops that span from micro to macro scale. So this is quite a, I think for an everyday person, difficult to grasp what I'm interested in. But luckily, through my artistic research, I found the medium which is very potent in metabolism, and this is soil microbiome, or soil where there is a lot of bacteria, archaea, and fungi. So fungi are not new in my artistic research, so since one of our works was actually a stage for collaboration between fungi and the human nervous system. It's called MyConnect, an interspecies connector, which plugged the human body in a feedback loop with fungi and thus questioned human separation from the environment and blurring the body-environment division. So a person who was lying in this capsule was actually exchanging signals with the fungi. Another process that I tend to research continuously is symbiosis. So this was a really nice experience also to meet Toby, because we were able to discuss a lot of different phases of symbiosis, I would say, and see how in different contexts can be understood. But basically how I understand it is like a negotiation, this ongoing negotiation, which is very much context and time dependent. In one of my previous works, Symbiome, the interspecies negotiator, we sonified the process of symbiosis between red clover, Trifolium pratense, and soil bacteria Rhizobia. The idea was to make fluctuations of symbiotic relationship audible and reveal the influence of the environment onto symbiotic relationship via sonic modulations. So the visitors were really able to hear how this is not a steady relationship, but all the time changing and exchanging, basically. With my works, I want the audience to have as much of an embodied experience as possible, since I want to convey and offer a bit of situated knowledge of another species, agent, or process. This means that the audience is not in the situation where they just read or see, but usually smell, hear, feel things. And in the work Inspiration, the breathing station that you're looking at now, visitors are able to inhale a bit of happiness. A happiness bacteria, Mycobacterium vaccae, which is distributed by the installation. However, the puffs of air enriched with happiness bacteria are not always evenly enriched by the respiratory masks. Inspiration acknowledged that an inhalation and exhalation are intimate processes that bind humans to their environment, but also asked what happens with the connection once it is technologically mediated. Who or what will dispense the dose if we will have breathing stations, and what does the dose contain, and who will survive because of this dose? So another thing that I'm interested in is interspecies relationships. And throughout my artistic practice, I consciously observe them and look at what kind of entanglement we have with them, with what kind of biopolitics and necropolitics comes in each relationship. And this politics defines quality of life for particular species and also their possibilities of survival. One of the artworks that deals with this is Transversal is a Loop, where I do a sound performance together in collaboration with crickets. And this is also a research of cricket-human relationship. And this work spans from sonic immersion into biological environment and then goes through a reality of crickets which are actually in the process of extinction, as many insects are. And then to one of the most feasible possibilities of survival in planetary metabolisms that are at work at the moment, commodification. So I'm here in the research was looking for a way to find how would cricket survive in the system that we have now in the ecosystem and capitalistic system, and the only answer I could find was basically through commodification. But commodification as the mode for survival, unfortunately at this moment is possible only from one species of crickets. And this is species Acheta domesticus, which is a standard feeder insect for pet and research industries. As mentioned before, planetary metabolisms are of great interest to me since they link the presumably unlinkable and reveal dependencies we are very much part of. In this work, Earthlink, the system of biological installations, aims to serve as an entrance point to the post-anthropocentric constellation of environmental relations and planetary metabolisms. Feedback looping entanglement connected with tubes circulating air saturated with organic and inorganic compounds entwine biomes with microorganisms of various origins and breeding station, a breath collector, and an ecosystem in constant negotiation affecting species from micro to macro scale. And now we have come to my latest interest, and this is storytelling, mythologies and worlding. I came to this because, of course, as an artist, I work with a lot of materials and I look at them and I give them meaning. And more, of course, I was aware of how I'm constructing the meaning for the material making. What is in front of me is just a bit of what this becomes when I talk about it. So that's why I was very interested in this. So I believe that any description or metaphor of realities expresses a bit of truth. However, this is not a one-way process since it also forms realities, brings them into being, it materializes them and co-creates them. Formation of metaphors is a two-way process where conditions for life of many beings are negotiated and at key moments also performed as abilities or disabilities, as support or negligence, as cooperation or as exploitation. The quick question in any of these stories or myths or scientific theories that we tell ourselves and others is what kind of world or realities or conditions are we creating with our stories, theories, interpretations? What kind of world are we evoking, building again and again? So as one of these examples of metaphors for fungi was Wood Wide Web. Is a metaphor for fungal networks that it's become very popular and widely spread in the past decade, I would say. However, there is something missing in this metaphor. Something is taken from fungi with this metaphor. It's a fungal agency. Fungi are not just entanglement of wires, as this Wood Wide Web suggests. It's not a system of inputs and outputs, but entanglement of a being whose individuality is very hard to even define because of these numerous nuclei that are flowing, these flows that Toby showed us. So fungi do make decisions, as Toby showed us, in which directions to grow and with who to share their nutrients that are not inert and inactive, but very active. So the World Wide Web metaphor takes away the agency from fungi, and this is very important to know when we're building myths, stories, and theories. We should be conscious that we might be taking away something from the organism we're describing, and with that taking a particular ethical stance. An ethical stance that will perpetuate a particular socioeconomical situation for both human and more than human beings, such as fungi, or it can start changing the world to be more inclusive, supportive, caring, and maybe wise, since truths are always complex and usually not so simple. Simple descriptions might actually be a sign of dangerous exclusion of multiple beings that might not be involved in processes of negotiating their conditions of life. So during my research, I came across this aphorism of George Box. The sentence is usually considered to be applicable to scientific models in general. So all models are wrong, but some are useful. And then I thought, what if we changed every once in a while this last adjective and thought of what about if all models are wrong, but some are caring, some are inclusive, some are speculative, empathic, harmful, or intuitive. what could that tell us for our research? So this was my last sentence that I want to put out there, to think about how when we do the research, what kind of models are we using and what are they saying? And in this year, I made the whole new series of works, which is in production. And now I would just like to show you a little teaser video of the series of works that are coming. So just bear with me and I will stop sharing for a second and start sharing again. Okay, and here we go.
Micro-mythological machines were assembled to tap into the underground flow, while the data poured out of it, along with the numerous stories. No matter where the flow was sampled, there the stories were, flowing out of the mycelium network together with much needed nutrients. There were many whispers, but some stories seemed the same as they trickled out of Haiti in chunks and pieces. Almost as a repetitive mantra that loops and loops. However, they were never completely the same. There were always glitches of differences, as if they would be assembled again and again, like some sort of distant memory, and then murmured by the machine.
We can't return to them because the notepad was precisely the problem. Was precisely the problem. We can't return to them. Was precisely the problem. We can't return to the normal. The problem. We can't. What we have was precisely the problem. Was precisely to normal. We can't because the normal was we can't return precisely the problem. Was precisely that we have. We can't return to normal. problem that we have that I can't return to normal. We can't have this precisely the problem.
This is the end of my presentation.
Thank you. Another fantastic presentation. I loved it. Yeah, and there are so many things that relate to your both presentations. So I'm also glad that you came to work in the 3-package deal with Toby this year. Very unfortunate that we had to cope with COVID, but I hope this is not one of the last things we do in this residency of yours, and I hope your relation with Toby goes on. To give a reaction that maybe leads us to a little bit of a discussion here. I think it's interesting that you, and correct me if I'm wrong, that you also advocate for even looking at with more understanding of the complexity at those interactions. Also looking at this last quote that all models are wrong, but some can be used for specific purposes, and those purposes can also vary. The other thing I thought was also very interesting that you said is about the cricket work. In a way, if you, as a species, do not allow yourself to be commodified within an ecology, you tend to become meaningless and isolated and die out. At the same time, you're also looking at archaea, which are these very slow growing organisms that grow on almost nothing. Would you plea for a world with only that kind of organisms, or do you plea for more complexity with the risk of some organisms being excluded from this very complex exchange? And so it's also, I'm looking at this idea of agency, and whether agency is something that you can, that also has the difficulty of being another reduction of the reality you're looking at. So that would be my question to both of you. Yeah, because in a way, if you look at this exchange being a marketplace, human society is more than just a marketplace. There's also people that give each other presents that go past the market. And I would be interested to know from both of you whether these models create a bit better understanding for your type of research.
Who goes first?
Go ahead, Svend.
Well, of course, I would say that there is much more than to any organic life than just trading and exchange. Maybe the problem that we have, or the challenge that we have to see different kind of economies in different kind of ways, because we are also used only to used or practice a very particular economies, but there are several, I don't know, different economies that existed before or still exist among, for example, indigenous people towards their environment, or some other ways to dealing with them. And then through that, we could see different kind of also interpretations of the exchange that is happening in fungi or somewhere else. So it's like always we could use more. I like the sentence because it doesn't give only one truth. It says there is more truth. And that's, I think it's very important to have somehow in the framework of the research. And then once you get that settled, you can be open to adjust your view and see other realities. And that's what I feel is very important, because then the knowledge can just accumulate and you can look at it through that. But that's the problem of complexity. Well, that's how complexity comes into the situation, and you have to think in a very different ways. And I think that's something that needs to be learned still a lot. So, but I still think.
Yeah, I think that Saša hit on something really important with that last sentence. Because by putting a different word at the end of it, that's exactly how evolution works, right? So it's all about trial and error, and not every model is good, and then all of these different traits that, I mean, it's just mind-boggling the way that evolution works, and all, this relentless testing of different phenotypes and genotypes, and to be able to be reminded of that also exists, for instance, in mythologies and stories. And I think that also came into play in the voiceovers in your videos, where it was talking about assembling and deassembling and assembling again. I think that's a view that we lose a lot as scientists. We like to focus on what's here right now and trying to explain the adaptive nature of that particular strategy or decision. But that's why I've enjoyed working with Saša so much, is that her work sort of reminds me that it's so much bigger than that, because this is just one point in time, and stories have been told about it for centuries, and it's the same kind of work that is being done in our lab. It'll just be a story someday, and some of it will be true and some of it won't be true. But just the fact that it's always changing, constantly changing, and that's refreshing. It's also scary, right? As a scientist, where you want to nail things down, right? You don't want to always be reminded that it may not work like that. So I find it both really exciting and humbling, I guess I would say, to sort of have that bigger perspective.
Yeah, could you say something about the more complex economic exchange that you find there? Huh? Because what you showed us is that if your fungi finds a route that is not delivering enough, it goes to another route, and that may be of another individual plant, but it may also be of the same plant. So do you sometimes encounter on the one side more neoliberal kind of exchange, so more taking, more extractivism, and on the other hand some more cooperation? Do you observe that?
We do observe that, and putting names on it, in some cases helps us describe it better. It also gets us into trouble, because then we try to pinpoint something that we see again in one point of time, and we don't know how long that strategy lasts. But we see all kinds of really sophisticated strategies. For example, we see instances where if you have a high nutrient resource patch, where the fungi will take it up, but then not trade it right away, right? It'll store it in these storage organs inside the hyphae, and wait until the plant is really, really low on nutrients, and then trade, and is willing again to pay more for those resources, right? I mean, if that's not neoliberal, I don't know what is. It's capitalism at its finest. But there's also cases where we see strains that seem to sort of trade willy-nilly, right? That don't show those same kind of sophisticated strategies. We do work, for example, where we'll take plant communities above ground and we'll shade one and let one grow in the sun, right? And then we try to figure out, okay, how does the fungi detect, where it's sending resources? And, on average, we find that it's going to send more resources to the plant growing in the sun because it's getting more carbon, but there's always outliers, right? Is that just noise, or has that fungi evolved some other kind of strategy that's that's a bit more socialistic? I think that we see a lot of range of strategies, and the more data that we gather, I think the better picture we'll be able to paint. But because this is all happening underground, getting those few data points can take years. So I think that's also something that needs to be said, is just the sort of the technical difficulties in trying to understand how these organisms make decisions, right? You can poke them, you can prod them, you can see how they respond, but it's really difficult to actually understand what input is driving those strategies.
Yeah.
I would just like to add about technical difficulties. Like, I was shocked to learn that it takes 4 months to grow mycorrhiza in a way that then you can maybe image a video. So it's like, a lot of things can go wrong in these 4 months. So that was for me quite, it's a big technical difficulty to do this.
And I think that's what makes it amazing. I mean, you know, we have all of these examples from science of, you know, fungi solving mazes and things like that. And I think that the thing that we're trying to do slightly differently is there's a big difference between free living fungi and these symbiotic fungi. And I think a lot of people don't really understand that difference. But imagine you're an organism where to be able to grow and reproduce, you are required to trade. They cannot live without plant roots. And so it's quite interesting being an evolutionary biologist and trying to figure out, you know, how do you even build a model whereby to optimize your own growth, you have to optimize the way you interact with another organism. They can't live on their own, and yet they're the most, among the most, if not the most successful organisms on Earth, right? And it's something like 80 to 90% of all plant species have these fungi growing in their roots. And the relationship is 450 million years old, right? This is an ancient ubiquitous symbiosis. They're very dependent on a partner, and yet that works for them. So that also, I think, says something about cooperation and dependencies.
Yeah, Toby, your research is quite fundamental research into biophysics of these types of roots. And root networks. Could you say something about the application of that research, where it may go? Does it teach us about living algorithms? Can we learn from that, of biocomputing, etc.? Can you say something about that?
Yeah, I think it goes from a whole range of applied issues, right? I think sort of the most immediate and important applied issues have to do with agriculture. These fungi are found in the majority of the crops that we eat in the root systems. They represent a very sustainable way of getting nutrients for the plants. And one of the things that we're working with is trying to breed a more altruistic symbiont, right? I mean, can you imagine, you know, this is kind of what we didn't do with crop plants that allows them to grow in groups and be less competitive. You know, can you select for a fungi that spends more time collecting phosphorus than reproducing itself? Can you know, one that's giving instead of always strategizing? If we can really push the relationship with these fungi in agricultural systems, then, you know, we can sort of move away from a lot of the chemical fertilizers. So that's, I think, the most immediate. I think sort of climate change issue is really big. These networks store an incredible amount of carbon below ground, right? So, I mean, I think it's something like the air and the plants combined still have less carbon in them than the carbon stored underground. If these networks die, we basically release all of our carbon, and, you know, the planet, we have to talk more about Planet B, as you have been working on.
But then, if it's so interesting to also get those insights from Saša's work about commodification of species, and at the same time, you do this research to research how this exchange between plant roots and mycorrhiza can become more altruistic.
Yeah.
Yet at the same time, the ethical question is also, how can we, as humans, as human society, become more altruistic and share that Planet B?
Yeah, I think it's all big questions. I mean, I'm really operating under the guise that these, you know, these fungi have been honed by natural selection to be as selfish as possible. And so, you know, as humans, can we tinker with that? But there's huge, you know, consequences to tinkering. But yeah, the key point is protecting these networks, I think. We recently started a conservation organization that's really focused just on protecting underground networks. because they are so vital to the health of our ecosystems, and they're so unknown, and there's so many very cool spots of endemicism where, you know, it's kind of like coral reefs underground where we have no idea kind of species diversity and interactions that they have with all kinds of organisms. So I think, you know, when you think about applied, we really want to understand them. We want to keep them alive and we want to know where they are. We want to map and protect them. And so that's a really big push in our group as well.
Yeah, it's very interesting. You should tell me later more about this protection initiative. You also mentioned coral reefs, which are interesting because they express so much more beauty, and therefore everybody thinks like.
I disagree.
Oh, no, but
more beauty than networks? No way. I don't think anything's more beautiful than these networks.
For the general audience, if you talk to the general audience, you say, yeah, I'm the president of the Coral Reef Protection Initiative. They all say, yes, this beauty going away. But can you imagine saying the same about your fungi?
Yeah, well, that's what we're trying to work with Saša. I think it's really, you know, wonderful to work with artists because, you know, looking at that last video, it makes people see these kinds of organisms in a different way, right? It's only because of how our science represents them that I think people think of them as non-charismatic, right, or not beautiful. I think that's why, you know, it's such an honor to work with Saša because it gives a totally different vantage point on what's happening. In these networks. So yeah, could you speak on that, Saša, a bit? Is like, do you think of them as ugly?
No, but since you know, since one of my first projects, I worked with them just because they seemed so beautiful to me. So it's just like to me they were always like the nervous system of the soil. So it's a different kind of nervous system, so it works there.
I didn't say they were ugly, huh?
Okay, you're not sure.
No, I agree. They are so beautiful. But it's interesting to hear you say that you need more artists like Saša to embark on that same research you are doing, not only for creating more understanding of the beauty of those underground networks. But also for the research questions that are in there. So I want to conclude here with a little observation, and it is, if you talk about the economies or an economic view, a market view on the exchange between plants and fungal networks, I think that marketplace in a way should also include us, maybe not in the direct interaction, but in the thinking. We're part of that marketplace in order to make that exchange on a global scale in Planet B more altruistic. So yeah, that's maybe very personal view, but I want to thank you for your presentations, both your presentations and your explanation afterwards. And yeah, I hope to you stay in touch. And that we see you, Saša, soon again in Amsterdam to learn more about the works of art you are developing as a result of the 3-package deal residency the last year here in Amsterdam and your work with Toby. So thank you. And yeah, we go back to wrapping up for this Kepler's Garden at Planet B. We hope to see you all again tomorrow. For another number of talks, and again, some work that Esmee Geerken is going to show us from the Amolf Center. So thank you. For those of you that are around, there are some drinks, and hope to see you tomorrow.
Thank you.
Thank you, Saša and Toby. Bye-bye.
Bye-bye.